MCM is, that is mini chromosome maintenance protein complex of this 2, 3, 4, 5, 6 and 7. These are 6 proteins, and they are combined together to make this complex. So how this its role played. Then there is a initiation complex. What we have seen is, and in that this cyclin dependent and this DDF dependent, that is DDKs, they are playing important role. It's kinases CDK and DDK, and they are playing important role. Then CDC45 protein, that is again important in making the complex. Another protein complex is GINS, which plays important role, and this is composed of 4 small protein like SID5, that is SID5, CDC105, PSF1, PSF2, PSF3, and it represents, this GINS represent Go Ichi Ni San, that is Japanese word, that means 5, 1, 2, 3. So that combine this form. So 5 dash 3 dash way it going, and in between 1 2 are there. So 5 dash to 3 dash, this is what the sequence is, and accordingly it is there. Then MCM10 is the different protein than MCM227 or this hexamer. Then important, we have seen that DDK and CDK kinases are there. I told you we shall be seeing it later separately because they are playing important role. These proteins are part of this GINS protein, and we have seen important. So next is how elongation takes place. That is, once this repliosome is formed, this elongation takes place, and accordingly the and accordingly whatsoever pre-replicative complex is there, from that there is a elongation that starts. The cells pass into S phase. Then we have seen earlier it was in G1 phase. Now from G1 phase it goes to S phase, and we have also seen that is the right number is a mathematical number. Then there is a supercoiling that takes place, that we have seen last time. Then topoisomerases are avoiding this kind of supercoiling. I told you one simple example or simple way of doing it. You try to pull the thread, both strands simultaneously, and you'll find that there is a coiling takes place because of the whatsoever wing structure is there, and that this topoisomerases play important role in that. Then next structure which is wing form is called as a replication form. I told you it is a comb like structure and or fork like structure, and this is the first stage, or rather from which it is starting. There are we call it as a junction between two strands, that is the leading strand and lagging strand. Okay, and then again it becomes this double stranded. This duplex DNA is anti-parallel. The DNA replication occurs opposite direction between two new strands at the replication fork, and all DNA polymerases, they can do the job only in 5' to 3' in better way, because it is in a smooth way. Okay. Now what happens is, there are what the other strand which is coming in opposite way, that is 3' to 5'. Okay, and when it comes, then it cannot be do it in continuous one. So it will form the haphazardly, it will form the breaks of that, because it has to do it in reverse. For it is, suppose it is just like a assembly, okay, as we have seen. So assembly has got a single track like that, and it becomes easy, then one by other part can be put easily. But suppose somebody has to go in opposite direction, then it will be difficult. So what it will be doing is, then some portions will be made, and then they will be made together. So then if it is opposite one, then it becomes a lagging strand, and this lagging strand means these are the part of DNA is made in fragments, and these fragments we call them as a Okazaki fragments. Okay, so these fragments are there. These fragments are then connected together to make a continuous one by using the enzyme. The enzyme like DNA ligases are there, and the leading one is a continuous one. Leading is a continuous one, and therefore people say that, or scientists say that it is a semi-continuous process. Why semi-continuous? Because one strand is continuous, another strand is in a haphazard manner, in a portion-wise manner. So therefore it is a semi-continuous process. The replication is a semi-continuous process. Okay, so these forks are there. This DNA is antiparallel. It occurs in opposite direction, as we have seen, and these two replicative polymerases, they synthesize DNA in a opposite direction and having opposite orientation. So this replication form, this fork that is continuously, and thus I told you that various polymerase enzymes, they are doing it. They are synthesizing DNA in opposite orientation. Polymerase epsilon that synthesizes DNA on the leading DNA strand continuously. So we call it as a semi-continuous. While the other one, they are doing it in a fragmented one, that is polymerase delta, which is doing it in a lagging strand, and thus it is making an opposite template strand. It is in fragmented strand, and these fragments which are made by polymerase delta, they are called as a Okazaki fragments, which are connected together. Okay, so they are Okazaki fragments are about 100 to 200 base pairs in the eukaryotic replication fork. This lagging strand usually contains longer stretches of single-stranded DNA, and these are coated with the single-stranded binding protein. Now this is very important in what sense actually, these Okazaki fragments. Why? Because we have seen that these are made in a fragments, and while they are made in the fragments, so there are some initially and some end portions are there which are removed, and then they are again connected together with enzymes. Like there are primers are there, RNA primer is there, and which has to be removed so that the only DNA and DNA parts are there. So the enzyme is there, like RNase enzyme is there, that is which enzyme causes removal of this RNA primer from that. And then lately this RNA primer, after removal of these portions, then only DNA portions are there. This flaps like structure it is making, and from that this ligases, as I told you, they are comparing or they are joining it. Okay, and it repairs the nicks which are left between these 3-hydroxyl and 5-phosphate groups of the newly synthesized strand. Now why that way it is important is this is a process which has got a chances of error, because the DNA strands are there. In that one RNA primer is there, that primer has to be cut off with the enzymes, and once when the primary has to be cut off, then some of the portion of the DNA if changes, then it can change whole sequence, and this becomes the one of the reasons for the mutations in the DNA at many places or many times. If something goes wrong, you can understand the complexity of the cycle. So during one DNA replication, it is says that around millions of times this fragment formation is going on. Okay, because these are so big strands. So if these strands are there, this process is so big. If anything goes wrong in that, that can result into fault in the DNA formation, and then this DNA fault formation can be result into any kind of pathology. So this is another one more reason or problem which is associated with the pathology. It may be observable pathology, or it may not be observable or recognizable pathology. If you remember last year pharmacology, there is a branch, we call it as a pharmacogenetics. In that we have seen that there are genetic differences are there in people, and because of that either their metabolism, either their receptors or their transporters have different structure or different protein composition, and because of their when we say that genotype is changing, there is a change in a phenotype. That means because of the change in the genetical structure, there is a change in the functional capacities. So these are the things which are the basis for the problem. Okay, so we have seen that there is a leading strand which is a continuous one, which is basically using the template replication form in 5' to 3' direction, which becomes easy because the DNA polymerase enzyme is there, that takes only portions only. So like that it will be there. So it is a continuous strand which has been used 5' 3', and therefore there is a continuous one, while in other case there is a Okazaki fragments are formed, and these Okazaki fragments are in lagging strand they are formed. So this newly synthesized DNA strand which becomes complementary to the original strand, which is to be synthesized in the same direction in 5' to 3'. Once RNA primer has been added by primase enzyme to the 3' end of the leading strand, the DNA synthesis will continue in a 3' 5' direction with respect to the leading strand uninterrupted, that is continuous one. Therefore I told you we call it as a semi-continuous process. DNA polymerase epsilon will continuously add nucleotides to the template strand, therefore making a leading strand synthesis require only one primer, because only once it is primed, once the RNA primer has attached, then it will continue without any break. While in next one, that is in lagging strand, everywhere we have to see that the primase is the RNA primer is attached, then it will go for some time, then it will break down, it will stop, that much fragment is made, and after that fragment again RNA primer will be going, this RNA primer will be making again new segment of the DNA. So many segments will be made, every time there will be RNA primer will be attached, and this will cause so many fragment to what we call as a Okazaki fragment. This Okazaki fragment will then connected together after removal of these RNA primers, and these RNA primers after removal of this all this connection, this will become a continuous one. So the first one was continuous, this is discontinued process, and therefore this replication becomes a semi-continuous one. So there are various polymerases which are playing role. We have seen them, that is polymerase alpha, then polymerase delta, polymerase epsilon, and they are playing role at different places. This polymerase alpha forms a complex with a small catalytic subunit and large non-catalytic subunit, and it synthesizes an RNA primer. It allows DNA synthesis by DNA polymerase alpha once at the origin of the leading strand and at the start of every Okazaki fragment, because this is responsible for making RNA primer. So in leading strand the RNA primer is once make, because it is then continuously happen, while in the lagging one, in the lagging strand, the RNA primer has to be attached every time. So every time it is attaching. So this is the job of polymerase alpha. We have seen polymerase delta playing role in the lagging strand, that is in or making Okazaki fragment. We have seen polymerase epsilon is highly processive. It has got a proofreading facility, means it will not miss any kind of single nucleotide into that. Okay, and it is mainly error checking with the polymerase delta. It is helping into that. So these are the various complexes which are formed. I'm not going to that much detail how each and everything is happening. We have just gone through various basic activities which are being taking place, and how these basic activities are playing role into this major DNA formation. Then lastly is the proliferation of nuclear antigen. The polymerases require additional factors which support DNA replication. These DNA polymerases have a semi-closed hand-like structure which allow polymerase to load onto the DNA and on this hand and it begin translocating. So this structure permits DNA polymerase to hold the single-stranded DNA template. It incorporates this deoxynucleic these triphosphates, which are the energy sites or energy molecules, and they release the double-stranded DNA. And structure of DNA polymerase does not allow continuous stable interaction with the template DNA. So this is how this happening. And for that there are to strengthen the interaction between the polymerases and template DNA. The DNA sliding DNA clamps, they are associated with the polymerases to promote the processively of the replicative polymerases. And there is a structure is there. It makes the structure a sliding clamp structure is there. A ring-like structure is formed that is known as a proliferating cell nuclear antigen, which has polarity with the surface, and it interacts with the DNA polymerases and then tethers, means slowly breaks into the DNA template. This DNA, that is this antigen dependent stabilization of DNA polymerases, have significant effect on the DNA replication because they are able to enhance the polymerase activity or its processing up to 1000-fold. It is very essential cofactor and has a distinction of being one of the most common interaction platform in the replisome to accommodate multiple processes at the replication form. So it is also viewed as a regulatory cofactor for DNA polymerases. So it regulates it, and if anything goes wrong, that regulation goes down. Then the replication factor C is there. That is again important, and this is that is the antigen is loaded by the replication factor C, and thus they are complementary to each other. Then lastly is a stalled replication fork, that is DNA replication at the can be halted, can be stopped for some time, or just like a pause button you have by a shortage of this deoxynucleotide triphosphate, which are the energy source, or by DNA damage, and thus it is causing a replication stress, and this replication is called as a stalled replication fork. The fork protection complex of the protein, they are stabilizing the replication form till the DNA damage or other replication problem are fixed. If it is stalled for a long time, it can further lead to more DNA damage, and then it can be progressively increasing. The stalling signal are deactivated if the problem causing replication form are resolved, then it is fine. So if you remember earlier class, then we have seen that DNA has got its own, or the chromosome, they have got their own repair process or DNA repair process when we have compared DNA, RNA and proteins, which is not there in a protein or any other thing. So this is how it tries to rectify as much as possible. But if this rectification mechanism is not working, then we can have the problems with the DNA or genetic problems which are related for or which are associated with various genetic disorders, or the basis of genetics in some of the pathophysiological or pharmacogenetic conditions. Lastly, how there is a termination of replication. It requires different processes depending on whether the chromosomes are circular or linear. Unlike linear molecules, circular chromosomes are able to replicate the entire molecule. However, two DNA molecules will remain linked together, and this issue is handled by decatenation of two DNA molecules by topoisomerases. These are also used to separate the linear strand, as they are intricately folded into a nucleosome within a cell. So they are having important things that they are having the particularly related to lagging strand. There is a disadvantage that in replicating entire chromosome, the leading strand can leading because it is a continuous leading one, so it can use a single RNA problem that we have seen. and multiple RNA problems are there in the lagging strand. They are Okazaki, that is also we have seen just now, and it leads to issue due to the fact that DNA polymerase is only able to add 3' to 5'. So in that case, what happens is these strands are made, and they will result into progressive shortening of both daughter chromosomes, and this is known as a end replication problem. So it is handled in by telomere regions, and telomerase enzymes, they sort out this problem, because there are, one is continuous DNA and other one is a fragmented DNA which is combined together, okay, and these are joined together, and if any problem is there, it is repaired. So this all things are there. This repair is done by telomere region of the chromosome. In chromosome you may be having, we have seen the various regions are there. Telomere means the last or the end region of the chromosome, and there are enzymes which are present, they are known as telomerases. So they are taking part into that, and they are having enzymes or important enzyme, they are known as a reverse transcriptase, or they are also called as a telomerase reverse transcriptase. We know transcription. Reverse transcriptase are the enzymes which do, that is RNA transcriptase, what they are doing, DNA to RNA, that is transcription they are doing. Reverse means RNA to DNA. So the RNA component of telomerase that will converted into DNA, and this is then joined, or it is helping in the repair of this process. So this telomerase reverse transcriptase will do the synthesis of DNA, and then it will be incorporated into that. This is how the repairing will be takes. Okay, so there are such things will be happening. The replication factories means there it occurs in a localized way in the cell nucleus. The replication forms are moving around in the standard DNA. The concept of replication factories emerge. That means the replication forks are concentrated towards the immobilized factory, and thus these template DNA strands, they are passed like a as I told you, conveyor belt like situation, that is which is happening in the industry. If somebody can do it, that is how motorcycles or the cars are made. So this is a conveyor belt factory, or at many places this is there, that is one after another actions are there, conveyor belts are there, and on this belt you may able to see the YouTube movies of these conveyor belts in automobile industry, particularly whether manufacturing. So this is how sequence of the things are there, how the assembling of this are there. So this is important, and thus they are taking part in cell cycle. So far we have seen how G1 to S phase it is moving, then slowly it will go into G2 phase. We shall be seeing all this into cell cycle when we shall be seeing cell cycle. However, during that there are various, we have seen already that G1 to S it goes when it goes for the synthesis part, that is when the DNA is being synthesized or new DNA is formed, or exactly replication is going on, and in that we have seen various important proteins and enzymes are playing role. We have seen very important protein which we say that we may discuss it again, that is the CDK and DDK proteins are there which are playing important role. So we have already seen this, and these kinases are playing important in various aspects. Then there are various cell cycle checkpoints are present throughout the course, and they will see that whether the things are happening as per the plan or not. Okay, just like quality checks are there, or even if again you will see the this conveyor belt facilities or of automobile industry. So everywhere the checkpoints are there, that if the tire is fixed, then whether it is fixed properly or not, what has happened. So at every place these checkpoints will be there, and that will determine whether the cell is progressed through the division entirely or not. Importantly, in the replication, the G1 or restriction, whether what will happen, that is there, whether it goes to G1 to S or go ahead, or it will come back from G1 to G0 phase. We shall see all these phases in next topic, that is cell cycle. Just So if it is not going ahead, it will go back to G0 phase, to what we call is a resting stage, and this they are prevented from initiating any round of replication, because this important machinery like MCM proteins, they are not expressed, or they are not allowed, they are not formed into that hexamer, and because of that it will not be there. Okay, but once it goes to S phase, that means replication begins, and if the replication continues or completes, then it will go to the next phase, that is G2 phase. So these are various checkpoint proteins are there. There are many checkpoint proteins. We will not go for this each and every checkpoint. We should just understand that there is a process which is doing the checkpoint, because and during checkpoints they will stop the cell cycle from entering the mitosis if there is any problem is found. So they will allow time for DNA repair. DNA repair will be done first, and after repair only it will allow to go into the mitosis phase. Okay, these proteins are also involved in the DNA repair pathways. They stabilize the structure of the replication fork to prevent the further damage. We have seen that it was halted because of the less energy and some other reasons. So again, these checkpoint proteins will give the signals, and they will see that it has been started again. They are essential to avoid passing down mutations to or other chromosomal aberration to offspring. Aberration means any abnormalities are there, any changes are there which is not normal one. So these checkpoint proteins, they are essential to this. So this is these stages are helping us to understand important things. What are these important things? That is how this process is being carried out, and how important all the stages are, and if anything goes wrong, that will make the impact on the DNA, and from that DNA the whatsoever daughter cells are made, on that it will be made impact, and this genetic impact will be carried forward, and because of this whatsoever daughter cells are there, their functional changes that can be there if it is not properly done. So if the repair is not done properly, if the checkpoints are not properly utilized, and if the checkpoints are not checking the things what has been happening, then it will be making a problem. Okay, so these various activities are there. These will be playing important role. That is there are various proteins are there which are playing role. That is example of these are known as serine threonine protein kinases. Okay, they are called as ATR. Then other protein kinases are there. Okay, these are known as ATM, and these are all some of these we shall be discussing when we are discussing. We shall be discussing about the various pathways. Okay, in these pathways there are important pathways are there, and they are playing important role in many activities, including apoptosis and other things. Okay, they are ATRs are involved, that is serine threonine kinase are involved in the arresting the cell into response to DNA double stranded break. If there are break, then this will be there. It is a important checkpoint partner, and it will then make the actions accordingly. Okay, so these are various signaling protein. I will not go for each and every protein because there is a very big list is there. We should just understand how it is there. Then again, this is also not important, much important for that, how the replication takes place through nucleosome. That is these nucleosomes we shall be seeing as a part of detailed genetic structure, and these nucleosomes are basic winding unit. That is, we have seen a bead like structure of histone and on histone these are bound, and the DNAs are wound like a string on a reel. Okay, this is a first reel, second reel, third reel, like that. There are various such moieties, and they will be doing it, they will be making the replication. Now again, prokaryotic and eukaryotic DNA, there are differences are there. I will not go again in all the basic details, but basically the eukaryotic is a more complex, prokaryotic is comparatively simple. Eukaryotic has got a very big DNA size, very long DNA chain is there. In prokaryotic it is small. In prokaryotic it is usually cyclic or circle-like structure is formed, while here it is a long and continuous process is there. So these are various differences which are important. You might have seen the basics in microbiology. Okay, we'll have the classic differences which are very typical one. So prokaryotic DNA replication occurs in the cytoplasm, while in eukaryotic it occurs in nucleus. Only one origin of replication per molecule is DNA. It has got many more origins of replication in each chromosome. So at various part, because it is a big DNA in eukaryotic, so this is happening at very place. The origin of replication is about 100 to 200 or that much nucleotides in the length. Okay, and each origin now here in prokaryotic it was only one. Here there are many, and each is having around 150. So you can understand how bigger one the eukaryotic replication is. Replication occurs at one point in each chromosome. We have already seen it occurring at several points simultaneously in each chromosome. So only one origin of replication, multiple origins of replication. Initiation is carried out by protein DNA and DNA A and DNA B, and we have got here it is a origin recognition complex, and many proteins are involved into it. We have seen various proteins. Here topoisomerase is needed. It is for that, and this is what mechanism of action we shall be seeing some of the antibiotic, that if those which are causing inhibition of topoisomerase, so DNA replication will not be possible. Okay, in both cases topoisomerases are required. In prokaryotic replication is rapid, here replication is slow because it is very big and it is very longer one. So there are various proteins, you can just go through the list, no details are required. So many proteins are required for this, and they are doing the job for the DNA replication. Okay, so here we complete the DNA replication from our syllabus. In the meanwhile, you can also go through, and if you have any difficulties, you can ask, you can have the questions on this. And tomorrow is a holiday. Tomorrow we will not have class. Rather, if time permits, then we may check, but most probably will not have a class. Saturday, Sunday, as usual, will not have a class. Monday I may try if I am free, because we have missed some of the classes because of the other activities I had. So we can see whether we can plan any class after on Monday also. So here we complete DNA replication. I am trying to make it as concise as possible, because that topic is still it is very lengthy one, but we are trying to make it as concise and as limited as it is. So I'll suggest you to keep your points ready, what's your taught, so that only that much is asked, will not go into more detail. Okay, so you please make the what we can say narrative or list of the points which are important. So take care, stay safe, and stay healthy. Okay, so we stop over here.